Okay, so we're going to keep talking about fungi. Again, apologize for the limited drawing ability with the last slide where we were talking about Pilobus. But the things I want you to remember about fungus is these things are pretty incredible. They're the first, one of the first organisms to actually move from an aquatic existence to a terrestrial existence. They can survive in crazy extreme conditions. They can go dormant and just hang out for long periods of time. So they're pretty darn amazing creatures or organisms, even though we look and go, it's just a fungus. So, all right. So here's another group, phylum, that I want you guys to make sure you're comfortable with. Glomeromycota. So let's put the red star, let me get back to red, and make sure we are recognizing this is a phylum of importance, one that we want to be comfortable with. Now, a couple key things to remember with these guys. Many members of this phyla are plant symbionts. They work together with plants. Sometimes you'll see these called the AM fungus. This is in reference to the arbuscular mycorrhizae. These are a structure that they form in association with plant roots where they're working as a team. So symbiont, in this case, many of them are mutualists where they're working together and they're both benefiting. So let me red star that because that arbuscular mycorrhizae is something we're going to come back to and talk about a little bit later. And I'm going to ask you guys which phyla of fungus is connected with the plant root, which one's the most commonly associated member or phyla with these symbiotic relationships with plants. And then the other thing to mention that's a little weird, at this point, we can't find evidence of sexual reproduction in members of this group. They're reproducing asexually, copy. Mutations occur which create genetic variation, but just not nearly the genetic variation you would get if there was sexual reproduction. So again, it's a little bit weird, a little bit goofy, but just feature this group at this point. As our knowledge grows, hopefully we find more information about Glomeromycota. Definitely an opportunity for somebody interested in fungus. Explore this phyla. You could be one of the first people to identify new traits and characteristics about this particular phyla. All right, let's now tackle the Basidiomycota, another big red star. Get this one under your belt. Make sure this is in your notes, and make sure you're good with all the information on Basidiomycota. So by far, this is the most common fungi. This is the one we consume, we use commercially. So those mushrooms you have on your pizza, Basidiomycota, toadstools, things called puffballs. So if you guys have ever been out in the yard or walking around and you step on a round mushroom and all of a sudden this little puff of looks like smoke comes up, that's a puffball. That puff of smoke, those are actually spores, so don't breathe them in. Shelf fungus, fungi, I'm just going to say etc. So the picture right here, this is a shelf fungus, or we can sometimes call them bracket fungus. Very common, very widespread. We have a lot of members in this group, and we have a lot of knowledge about this group because of their economic importance and value to us. They're also important plant pathogens, and this would be in a negative sense. That's not a upside down smiley. No, there we go. This is a problem. When you have fungus parasitizing your plants, you get a fungal infection on a tree, it can kill the tree. That's not a good thing. It gets into agricultural aspects. There's huge problems with this particular group. So again, why we invest a lot of time and energy into studying Basidiomycota. So the key defining feature, how do we know if a fungus is a member of this particular phylum? We look underneath it and we look for a structure called the Basidium. Now the Basidium is this club-shaped reproductive structure. So you take a look at your general mushroom. You got the stem or stalk here, and I'll go through the parts in the next slide. And then you have this thing called the cap. So think about the mushroom cap. Underneath it, these little lines here, those are known as the gills. So if you flip it upside down, now we're over here, and you look in these gills, the gills have hundreds and hundreds of these things called basidium. It looks like a little club. So that little club-like structure there. And within the basidium is where we're going to see the reproductive spores being produced. So those spores get released, spores germinate into negative strain, positive strain spores. They grow. So these are the little hyphae that will become or form what we call the mycelium. And then two of these spores can get together. Negative strain, positive strain can get together and fertilize and create the secondary mycelium here, which continues to grow and develop hyphae, gets bigger and bigger and bigger. It's spreading through your lawn. And then it says, oh, time to reproduce. Let's push out this mushroom structure. We could officially call it a basidiocarp. That gets bigger, and within the basidiocarp, underneath are basidium filled with spores. Okay, so that is the big key feature. So Basidium, this is why we're in the phylum Basidiomycota. They have a basidium. Okay, so here's the bigger, better, more microscopic view of these structures. These are the gills I was referring to. So take a mushroom from the grocery store, flip it upside down. You're going to find all these little gills here. You really want to look into it, throw it under a microscope. Crack it in half or cut it in half, put it under a microscope, and look for some detail. Microscopically, if we zoom in enough, we're going to see all these little fingers here with these little extensions and those little tips. Those little tips are where all of the spores are produced. So a lot of reproductive potential here. This is an example of a puffball. These are orange. A lot of times the puffballs in our region are white. And when you step on it, so it looks like a little cloud of smoke. Those are spores. Please don't breathe them in. That's not a good thing to be breathing in fungal spores. But here's your general structure. You got your mycelium down here, or hyphae, all the little tendrils of the hyphae. The part that's coming out of the ground is known as the vulva. So that's this structure here. Emerging from the vulva will be the stipe or the stem. As it grows longer, this little annulus, this ring gets carried further up, and then you have the cap up here, the pilus or the cap. There's our little mushroom cap. And on the underneath side of the mushroom is where you're going to have all the little gills, or laminella, if you want to use a fancy Latin for it. Gills is good enough. So, all right. So, general structure of a Basidiomycota. The basidium, again, is our defining feature and the important structure to remember. Make sure you're good with Basidium. Life cycle wise, we can start over here. Let's start with the strains. Positive strain, negative strain come together, form a mycelium. But here's that weird term, dikaryotic. The strain comes together, but the nuclei don't fuse. So you have two nuclei inside a single cell, N plus N. So the nuclei do not fuse to become 2N. They are sitting there as N plus N. So that mycelium, this is officially the secondary mycelium, will grow, get bigger, push up the Basidiocarp. That's the mushroom that we see coming out of the ground. And then that Basidiocarp inside the gills will produce the Basidia, which are still N plus N. Those Basidia will then make the spores. They're going to produce all those little spores that will eventually over here be released, and we call them basidiospores. They are N, haploid, half the genetics. Spores get released, they land in whatever the environment is. They may be blown away by the wind, they may be just dropped a few inches from the parent mushroom, the parent Basidiocarp, and then those spores can germinate into the different mycelium, positive and negative strain, and potentially they merge back together to create the dikaryotic one again. Okay? All right, so now that we have the general concept of how the Basidiomycota work, do their thing, reproduce, etc., I want to show you guys an example of a crazy one that odds are you have seen and may not have recognized. So I want you to look for these things. Best time of the year usually is in the spring when we start getting warm and there's still rain and moisture. Winter, fall, winter, not as good. Summer, depends on how hot and dry it gets. But it's a thing called a fairy ring. So Basidiomycota some members of it can form what we call fairy rings. These are literally circles of basidiocarps, circles of those mushrooms, and they just get bigger and wider and wider and wider as the circles grow outward. But take a look for these. They can pop up literally overnight. They might only be there 3 or 4 days, and then they reproduce, drop spores. The basidiocarps are done, and new generation is underground. The mycelium is always underground as long as environmental conditions don't destroy it. But take a look for these things. Do a little scavenger hunt. Go see if you can find a fairy ring if the environmental conditions are right. So, okay, on to the last phylum, Ascomycota. Big red star on this one. So, Ascomycota makes up about 75% of the known fungi out there. Common examples are things like yeast. Oh, let me get rid of that. Yeast. So if you're making bread or pizza dough, you're using fungus, Ascomycota. Common mold. Go look in your shower. Along the edge of the windows, cracks and seals, things like that. Cup fungi. Probably don't see a lot of these out there, but if you look, they're out in the woods, areas of kind of damp, moist areas. Truffles. If you got enough money, you probably have eaten a truffle. Not real common in this area, but more common, and here's a great opportunity for anybody interested in making money, morels. Morels. I've seen them go for $40 to $50 a pound. Go check out the park. Go check out wooded areas. Figure out what does it take to grow morels. Can you grow them? If you can't, can you find them? Do you know how to look for morels? What time of year? Where do you look? What trees do you look around because they're decomposing dead trees? But morels, incredibly valuable. Just in the Sangamon County, Menard County, you can find plenty of morels, and there are people who will go morel hunting and sell their morels. Ascomycota are also, unfortunately, some nasty plant pathogens. So let's give it the unhappiness. Huge loss in revenue agriculturally if you get these things in your fields and they start impacting your crops. So, all right, I'm going to pause it here. I like to keep these around that 15, 16-minute window, and then we'll pick up the rest of Ascomycota in the next lecture.